Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh and a very good afternoon. Welcome back to our last lecture in 2021. Next week we're gonna have our own new course for the new year. All right, so without further ado, since last week we, well, because of the flooding, so we didn't do any live lecture. So I did pre-recorded. So what we're gonna do today is I'm just gonna ask you guys briefly on the topic that was covered last week. Do you have any issues? If you do, you can raise your hands and ask now. Otherwise, I will just start. Anything? Because I'm not gonna cover it again for this week's lecture. Hopefully we don't have any more flooding and that I'll be able to complete all the lectures by next week, hopefully. Okay, and then we'll probably do our second quiz on week 13 so that on week 14 we can do a discussion on it. Okay, otherwise if you do have any questions or if you think that you have so many assignments or alternative assignments that you need to do in week 13, let me know so that I can still put the quiz on week 14. And then instead of having a discussion, what I will do is I will just do a pre-recorded lecture, so to say, on the quiz so that you can just view it on your free time. Okay, so no questions. I'll give you guys another like 30 seconds. If you want to ask anything, just either unmute straight away and ask, or you can just raise your hand first. Either is fine. By the way, you can hear me clearly, right? Right?
Yes, doctor.
Okay, cool. All right, so since there's no question, we'll just move on to the topic that we're going to cover this week. Okay, so we are still doing enzymatic technology, isolation and purification. So what we've covered last week is we've looked at like a general overview on how do you actually purify a protein. So and we've went down up until, if I'm not mistaken, until here. Okay, fruit extract. So today what we're going to cover is we're going to cover every single thing from buffer precipitation as part of the purification process, and we're going to look at some other techniques that you can actually use to kind of like substantiate or as an add-on to purify your protein. Okay, so buffer precipitation is a technique. I think I've stopped this on this slide last week. It's a technique by which you can precipitate out your protein from your cell. So originally you have a cell, your cell is expressing the protein that you asked it to do, and then you homogenize it, you centrifuge first run to just separate all the debris and your proteins and DNAs and whatnot. Okay, and then you take the solution, you conduct a buffer precipitation. Okay, so what you do in this process is basically you are precipitating out your proteins from your bulk solution. So again, you have a layer like that. So that one is your cell debris mostly, or cell membrane. Okay, and then the top layer is normally your DNA or RNAs, which is the lighter molecules, and then you have your proteins normally in the middle. Okay, and normally all of these, if you have not seen any centrifuge product of a protein, I have it here. Okay, so this is how it will look like. So you will have your, of course this is not the real example. This one is after you first centrifuge, but what you do is, what you normally see is you'll have a solution like this, okay, and a solid precipitate. Okay, so this solid precipitate, for your first centrifuge, you will still see a solid precipitate, and this solid precipitate, normally you just throw it out and you just take your solution because this solution contains your protein. Okay, so so you have your protein over there. Okay, as shown previously, it's in a solution form. And then what you do is you add in salts. There's different salts that you can use. One of them that is widely used is ammonium chloride. Okay, so what is the function of this ammonium chloride? It is to stabilize the protein in a solution. Okay, not to stabilize, to precipitate slowly by reducing the forces and hydration layer. So when originally, without having the salt put in, the protein, because it's in the solution, so meaning that it is actually stabilized, it does not precipitate, it kind of like dissolves in the solution. It's not purely dissolved, but it's stabilized in the solution due to the repulsive electrostatic layer and the hydration layer surrounding the protein. Okay, so it's kind of like when you learn about Stern layer, where you have layer, you have a layer like that. So if your layer is cations, then your first Stern layer, you'll have a negative. So it's a stabilized interaction. So similarly, what happened here is the protein is stabilized in the solution, and for you to actually precipitate it out, you need to destabilize the layer. And one way to do it is to add more salt. So if you have more of these salts adding in, then the salt will stabilize or destabilize the Stern layer, thus precipitating your peptides or proteins out of the solution. Okay. Promoting nucleation process, and then the particle starts to grow, and everything will precipitate out. But some of the, depending on the concentration, you might not see it's actually precipitating out. It might be just like what do you call it, nanoparticles floating around in the solution. It's there, it does not dissolve in the solution, however it's still there, it's floating. Okay, so one way for you to actually extract out everything, you can again centrifuge, as what you did in the original, when you first homogenize the solution, you centrifuge to take all the debris out. Okay, similarly, what you can do here is after you add in your precipitating buffer, you can actually centrifuge again to get the highest concentration of peptides or proteins as possible. Okay. And you can also repeat this technique. So it doesn't, you don't just do it once, you might want to do it multiple times. Why do you want to do it multiple times? Again, when you think about it, okay, your cell does not only contain your protein. I did that correct? Okay, it does not contain only the protein of interest. It contains a lot of other proteins that are, well, as part of the cell component. Okay, so when you precipitate it out, you do not actually precipitate out just the specific protein of interest. So you might also precipitate out some other proteins. So whether to know whether you actually precipitating out every single thing, you need to do like analysis lah. Okay, either a HPLC analysis, or for biology, normally they will do SDS page, okay, so part of the analysis. And then from there, you can actually identify what is the other proteins that is available in the solution. Okay, if it's still a lot, then you might want to repeat the whole process by adjusting either the pH, you play around with the pH, or you can play around with a different buffer solution to try and precipitate out your own protein of interest only. Okay, so you can repeat that, or additionally, once you have your protein, you can do additional techniques to try and purify so that you can get a more pure product. Okay, so that's why when you after a buffer precipitation, you don't normally call it a pure product. We normally call it a refined extract. Okay, an extract is a raw, very raw material. You have a lot of other junk in there. Buffer precipitation is part of your purification process. So, but so far, based on my experience and based on the papers that I've read, nobody actually managed to just do buffer precipitation once and manage to get a pure product. Okay, so normally you will do multiple times. Either you combine buffer precipitation And then depending on the quantity of your product, if it's quite low, then you might want to do just SDS page. And then from the SDS page, you can actually select the band of your protein, and then that is your final product. Okay, so that's one way to do it. Or if you're doing a bulk solution, okay, especially in industry where you normally don't just do a very small quantity, you do a huge quantity. So you might want to repeat this process, or you want to combine it with a different purification techniques. Okay. And then you can also concentrate the solution so that towards the end of the day, you get a high concentrate protein. And thus for an industry, that is what you want, because you don't want to, send to your via a big flask like this containing your proteins. You want to send them as small as possible, probably in the size of this cup. Okay. And then, it has a high concentration or high amount of protein. All right, so buffer precipitation, you can either, if you precipitate out the protein, of course, your protein will be in the precipitate, okay? However, you can also do the other way around, by which you might know that your protein is very, very soluble in water. So you want to play around with the solubility of the other proteins. So you precipitate out the other proteins and then leave your protein inside your solution. So that's one other way to actually do it. Okay. So either or, even if you do have a precipitate, towards the end of the day, you're going to collect your precipitate, you're going to dissolve it again, and then that is when you get your defined extract. Okay. So this is one example on a protein process. So this one is production of rice bran proteins concentrate. So similar concept, you have your rice bran. So over here is a rice, but the example that I give, especially because we are looking at biotechnology, we normally call it a cell. Okay, but again, rice bran is a plant cell. It's the same thing. Okay, so you do pH adjustment, and then you can do, you need to break the cell. So in this case, you can either use a stirring, cold milling, okay, you can use enzymes, ultrasonicator to break it. Okay, so we actually touch on this as a physical way to break the cell. And then you centrifuge, again, to separate the solids and the area or protein of interest. Okay, so in here, it says collect supernatant. Supernatant is the solution. All right. And then where you have your protein precipitation. So that's one of the process. So you do a protein precipitation and then you centrifuge in this case, because you can see the volume is quite big, huge. Okay, so you centrifuge again, then you get your solid and the solution. And then from here, because it says it's freeze dry, so most likely your the protein of interest for this product is in the solution. So you freeze dry it, then you get your protein concentrate. Okay, so that's one example of doing it. And again, because this one is not actually looking at a specific protein, so it's just a very general rice bran protein. So it must be a collection of proteins. It's not one specific protein. But in our case, because we are focusing more on enzymes, for example, to have an enzyme that have an activity. So you cannot have the enzyme as a mixture, because if you do, the mixture might be an inhibitor. For example, we will touch on inhibitor in the next few slides, or perhaps the enzyme will be inactive when it combines with some other stuff inside your a different protein, for example, inside your mixture. Okay, so that's just one example. If you, of course, this one is not listed in your lecture notes, but if you do want to have a look at that paper, you can just go to this address. Okay, of course, you can review this address later on, because I'm going to put up the lecture notes afterwards, or lecture recording. Okay, so once you have your refined extract, sometimes this is not enough pure for biotechnological purposes. Therefore, you want to, as I mentioned, you want to purify it again using a different technique. Okay, so these are some of the other techniques that I've listed. Am I still on? Okay, I am. Right. Okay, so these are some of the other techniques that you can use. We're gonna just touch about it in very brief. So you have, I separate them into 3 different techniques. Of course, there's more than this. Okay, because if you can see, HPLC is not as part of the list here. But so this is just an example. So number 1, we have a molecular size separation where you can actually separate or purify your protein based on the size. Some techniques, the name of some of the techniques belonging to this kind of like technology is ultrafiltration, dialysis, there's ultracentrifugation, okay, and this is common for neutral proteins, and industrial wise, this is the most common used technique. Why? Because this is the easiest technique that you can use, or that you can use for a bulk solution. Okay, remember in an industry, you don't just do it in a very small flask like this, okay, but you actually do it like in a very huge, like 200 or 300 liter tank. So it's a very huge tank. Okay, so centrifugation or ultracentrifugation is one of the easiest technique that you can actually use to kind of like purify your protein. Okay. Additionally, you can also use affinity technique. Affinity technique is more or less like a it's not here, ion exchange. So the principle is more like an ion exchange. Now my picture is not moving. Okay, I'm still moving again. All right, so it's more or less like an ion exchange whereby you have an affinity. So if your protein is a cationic, then we will use an ionic resin, and vice versa for a different charge. Okay. All right, and the other techniques is more on non-chromatographic. For example, liquid-liquid extraction, or you can use solid phase extraction, or you might want to use magnetic purification and whatnot. Okay, so even the magnetic is put there as well. So there are multiple ways by which you can do it. Oh yeah, I put nanoparticle under affinity because it's attraction between the magnets. However, it is also a non-chromatographic technique. Okay, chromatography is normally when you have a column and then you put in your sample and then it flows through and there's an interaction between your column. But the magnetic purification is a slightly different technique, and we will see it in the next few slides. Okay, so first off, let's look at the molecular size separation. As I mentioned, those are some of the examples. So over here, the basic principle for this molecular size separation is for you to have a membrane, okay? A membrane that allows a specific molecular weight to pass through it. Okay, so you extract out the exact size. It's not really an exact. So basically, you extract out the anything that has a lower molecular weight than the indicated on the filter, for example, a membrane, okay, using a distinct membrane. So over here, this is where, this is an example. There's 2 examples. The left-hand picture is one example, and the right-hand picture is also another example. Okay, so the one on the left is showing you a more a technique where you have a membrane, and at the same time you use a centrifuge. Okay, so this is kind of like a hybrid between a normal filtration and centrifuge. So you can consider this as ultracentrifugation. Okay, where without the gravity force from the centrifuge, your sample will not pass through the filter. Okay, so if you look at the filter itself, it says that 100 kDa, so meaning that 100,000 Dalton. So anything that has a molecular weight of less than 100 Dalton will pass through the membrane, okay, during the centrifuge. Okay, so you can see here initially, you put it in, you shake it to get a more uniform solution, and then you start your centrifugation. And then what you will see here is you will see a liquid at the bottom, and this liquid also contains either proteins, or for example, if you have your DNAs, your RNAs mixed in your solution, okay, it will pass through the membrane. Okay, but your protein will be up here. Okay, your protein of interest will be at the top of the membrane. All right, And of course, this all depends on the size of the membrane itself. If the membrane is 5,000, then anything that is above 5,000 will stay at the top layer, okay, and everything else will be pushed down and collected into your conical tube. Okay. And of course, towards the end of the day, you can add in more solution to make sure your protein dissolve properly, it doesn't precipitate out, before you can actually extract it out and use it for whatever purpose that you want. Okay, especially normally once you extract out, if it's an enzyme, you want to quantify the activity of the enzyme before you can actually sell it. Okay, so that's how in the industry work. Additionally, what you can do is use a dialysis bag. Okay, so I did not mention about MWCO. So MWCO is molecular weight cut off. So similarly to that one, so the 100 kilodalton that is listed here, this is actually the MWCO. Okay, so that's the technology. Normally, if you are using a dialysis tube or if you use any membrane, normally there's this terminology, MWCO, or sometimes it will just mention cut off. Okay, cut off 100,000 kDa or something like that. Okay, so the difference between these two techniques, the one on the left and right hand side is the centrifugation. So on the left hand side, you need to have a centrifuge to actually push through all the other molecules that is below 100,000 kilodalton to pass through the membrane. However, on the right hand side is more on a passive diffusion, where you have your membrane with a specific molecular weight cut off, and then you just dialyze it for 2 days or 3 days to extract out or to diffuse out all the other proteins, salts, for example, and some other stuff. Okay, and normally you keep on changing the buffer, or normally it's not just buffer; it's just milliQ. But you might want to change it to a different ones depending on the purpose of the dialysis. Okay, so you change it multiple times to make sure that the diffusion process is as at the optimum condition as possible. Okay, all right. So that is an example of a dialysis tube. If you have seen it before, that is quite blur. Normally, it's written there molecular weight cut off, and then what's the value. Okay. All right, so next one is affinity technique. It's more or less related to affinity chromatography, and the most common ones that is being used for biotechnology, especially for protein purification, is what we call as HISTAC. Okay, HISTAC proteins. What does it mean? It means that, so say for example, you want to express a lot of insulin. Okay, a protein example, and insulin. So what you do is you don't just so what you do is normally you will do a genetic modification on your cell. Okay, either you genetically modify the cell straight away, or you might want to use what we call as oh my god, I can't remember the terminology on top of my head. Anyhow, if I recall it, then I will mention about it. But it's basically a circular DNA. So normally you know that your DNA is inside your body, inside a nucleus, right? So what you can do is bacteria has two types of genetic materials. So some of them or all of them carries nucleus, same as what we are carrying. Some of them carries this circular DNA. Okay. That's the name, plasmid. Okay, so plasmid carries DNA, and scientists or industry, normally what we do is we don't alter the genetic of the nucleus of the cell, but rather we include plasmid, because plasmid also contains DNA. It contains molecular information, and you can actually, because plasmid is normally smaller than our DNA. DNA nucleus is very, very huge. But this plasmid can be very small, or it can also be huge. And the beauty is you can play around with plasmid. So it's actually rather easy to play around with plasmid. So you can modify the genetic of interest. And what you do is, say, for example, you have a sequence, DNA sequence of your protein over there. What you do is you just add in his. His is one of the amino acids, histidine. Okay, you add a polyhistidine at the very end of the protein. Okay, so once your cell express the protein, it will express the protein plus a tail or histidine tails. Okay, and this histidine tails can be used for in affinity chromatography to actually, well, I'll just talk about it in the next slide. Okay, so the principle behind it is more or less like a metal coordination complex, where the histidine actually binds to a nickel column. A nickel column, we call it as nickel column, but the nickel is actually bonded to a resin. Okay, and then you can actually purify it. The process will be shown in the next slide. Okay, and additionally, you do have a non-chromatographic techniques. For affinity, of course, LLE is not part of affinity. SPE probably, okay, depending on what type of resin that you are using. Okay, and of course the magnetic ones that we're gonna see in the next few slides. Okay, it's a specific separation technique that enables high quality purification. Okay, if you were to compare with the precipitation methods just now, as I mentioned, you can precipitate your protein of interest. You can also precipitate other proteins. Okay, however, for example, for histidine tag proteins, only proteins that has histidines, multiple histidines, can be purified. Therefore, we call it as a high quality purification. Okay, but why don't you do it in the very first place? Well, because for once, histidine histag purification is expensive, and thus buffer precipitation is cheaper. So if you were to purify like a very huge quantity, buffer precipitation is the best option. Okay. Interaction of the target enzyme with polymer bound, a reversible inhibitor. Okay, it's kind of like an inhibitor. We will look at it in the next few slides if we do have time. Otherwise, we'll look at inhibitors next week. Okay, but why do we call it as an inhibitor? Because number one, it does not damage the protein. Okay, uh, in contrast, if you were to try and precipitate out using by changing pH, for example, if you accidentally go into the extreme end, whether it's too acidic or too basic, yes, you can precipitate the peptide or proteins as you change the pH. But if you enter the extreme ends, you might actually hydrolyze your protein. Okay. However, if you use affinity technique, most of the time it will not damage your protein. Okay, so it bound but does not activate the catalytic process. Again, if you were to think about histidine tag does not participate in any reaction. So kind of like, you have a protein there, but it doesn't do anything. Okay, you kind of like you purify it. And then you can recycle it. So you can regenerate your nickel column. Similarly, and uh, when you are doing your ion exchange chromatography, right? If you recall in your second year, oh wait, you didn't do it. You probably do it online. But the same principle applies. So ion exchange chromatography, you can regenerate the resin, the embolite resin, so that you can reuse it. So over here, it's the same thing. If you have a nickel column, you can actually regenerate the nickel column, and then you can recycle and reuse the column. Okay, and more specific to protein than peptides. Again, why is it more specific to proteins than peptides? Because protein is normally huge, and of course, if you have your peptide with histidine tags, then you will still interact. But this technique is mostly used for protein purification. Because peptides, sometimes it's very easy to just use HPLC to purify it, so you don't really need to use a high tech expensive histag protein techniques. Okay, so this is how you actually this is how histag, the principle of histag works. So initially you have a lot of proteins, so target analytes. You put it in, you have your histidine, or you have your nickel column over here, and then you have your histidine. So for example, if the histidine tag is the one in red, it will have interaction with your nickel column, and then finally you can elute it, and then you get your pure protein. Okay, so this is how nickel column works. Okay, where you have a coordination complex between your resin and your metal and your histidine. Okay, so this is the side chain of histidine, and you can see here you can have multiple kind of like attachment between your polyhistidine and your nickel resin, and then you can simply use just imidazole to kind of like elute out all your histidine proteins. Okay, so that's the basic principle. Next one is we're going to look at a magnetic purification technique. So this is a rather new technique developed by this company, Takara Bio. Okay, you can have a look at the link if you are interested in looking at how they actually works. But basically, the general principle is, okay, you have this MAC-VGen. So MAC is just magnetic protein A nanoparticle. So you incubate it with your sample, okay, and then your sample will have an interaction with the magnetic component, okay, and then finally you can just simply put a magnet on your, say, vials or whatever to actually attract all the magnetic nanoparticles. And because your protein has already attached to the magnetic nanoparticles, everything will stay there. And then if you do your rinsing, you wash the solution, towards the end of the day, you will get your pure proteins. Okay. In this case, the example that is being used is antibody, but of course, if you can use it for antibody, antibody is also a protein. You can use it for any other proteins. Okay. So very basic principle, and useful, I guess. I have never used it before. I've used nickel column previously, but I have not used this magnetic thingy. Okay. All right. So I think that is all for today. Oh wait, that is all for just isolation. What we're going to continue now is we're going to look at the application of enzymes in industry, and to be more specific, we're going to look at the principle of inhibitor. Okay. Of course, if you do have any questions with relation to isolation and purification, you can either type it on chat, unmute, ask me now, stop me whenever. Okay. So we have about 20 more minutes. We're going to touch or start a little bit on inhibitor. All right, enzymatic interaction and inhibition. So as we've seen previously in Michaelis-Menten equation, enzymatic reaction occurs when enzymatic catalyzes a reaction. Okay, so you have enzyme, simple equation, you regenerate the enzyme, and you have your protein. So this is when you have the most ideal condition, whereby there's no inhibition, and there's nothing else that can stop or interacting with your enzyme. So you always have a high production of your product. Okay. The enzyme actively participate in the reactions, lowering the activation energy, as all catalysts do, formation of temporary bond, the ES complex, and some enzyme requires a cofactor and coenzyme, as we also looked at. Okay, so again, this equation that is written is the very basic equation. We do not consider the presence or the requirement of having a coenzyme and a cofactor. Okay, but as you see, as was shown previously, some enzymes do need to have a cofactor coenzyme to actually work properly. Okay. All right. So chemically, the application of enzymatic reaction is very straightforward. Okay. For example, if you're looking at P450 enzyme, in quiz number 1, you already know that P450 is an oxidizing enzyme. Okay. So it oxidizes your target. So in this case, for example, what you'll do is you have an OH group over there, you oxidize it, then you'll have this structure over here. Okay, you have a kind of like benzoquinone structure. Okay, and of course you will have a benzene as one of the byproduct of this reaction, and also if you looked at the indole aniline structure down here, what are the catalytic process that is being done by P450? You can simply just break that bond over there, okay, and having a indole and aniline. Okay, indole has that particular structure, okay, and aniline is having a benzene with a amine group. Okay, so that's one basic structure. However, enzyme activity can be influenced by formation or presence of inhibitor. So if you do have inhibitor, the catalytic activity either can be slow down, stopped, okay, either slow or stop, and then each slow or stop can either be a temporary slow down or temporary stopped, or it can be a permanent slow or stop. So there's 4 options that can happen if you have an inhibitor, okay, and we will look at some of the examples in a bit detail. Okay, so there are 4, or over here we're just going to cover 3 types of inhibition. So you have a competitive inhibition, you have a non-competitive inhibition, and the third one is you have a uncompetitive inhibition. So of course, if you look into enzymatic reaction and into inhibition, if I'm not mistaken, even now there's more subset of these. So you have like a huge different types of inhibition. Okay, but this is just an example. Okay, a competitive inhibition, it can be reversible or non-reversible. So how do you determine whether it's reversible or non-reversible? It depends on the reaction that is occurring. Okay, so for example, if the reaction is just doing a hydrolysis, okay, if you want to block the enzyme, you might want to use something that can permanently bound to the active site of the enzyme. Okay, one good example of a non-reversible inhibition is when you have or when you consume antibiotics. Okay, a penicillin G, for example, I don't have the structure. If I recall correctly, the structure is something like this. Okay, 6. Okay, so this is the basic backbone for penicillin. So this, wait, is it nitrogen or sulfur? I think it's sulfur. Okay, so this structure, as you know, of a square structure is very, very reactive, and this is what is found in our penicillin, okay, one of our antibiotics. And what it does is, if it goes into a bacteria enzyme, okay, so the enzyme of the bacteria function to one of them at least, to build up cell wall, bacterial cell wall. So what it does is this molecule, the antibiotics, went into the active site of the enzyme, break this bond, and form a bond like that to the enzyme. So it forms a full bond, and then this bond is rather unbreakable. So you do get a non-reversible inhibition. Okay, so antibiotics normally, most of them are non-reversible. There are some that are reversible, but most of them will go towards a higher end of non-reversible. Okay, so non-reversible or competitive inhibition, so to say, targets the active site. So if you have an enzyme like that, okay, and then if the substrate is a circle, the inhibitor might have a structure like an oval or a cylindrical shape, as long as it can fit inside the active site like that. Okay, now it becomes a competitive inhibitor. Okay, why do you call it competitive? Because both of the original substrate and the inhibitor are actually competing for the same active site. Therefore, you call it as competitive inhibitor. As simple as that. Okay, and then you have a non-competitive inhibitor. So we've looked at a competitive. Competitive means that it tries to go into the same active site as the substrate. However, for a non-competitive inhibitor, okay, again, if you have a substrate like that, a non-competitive inhibitor might actually bind to a different site. Okay, it binds to a different site. However, as you have seen the dynamics of a protein. Okay, the protein is not a standstill. It's not like a solid physical form. It fluctuates, it moves, it's a dynamic structure. So if you have a non-competitive inhibitor binds to a non-active site, what might happen is it might change the active site. Okay, so the active site might be there, but it might change the active site. Therefore, the original substrate can either no longer bind to the same active site, or the active site now becomes either slightly smaller or slightly larger, that it affects the activity of the enzyme. Okay, so therefore, a non-competitive binds to a secondary site and it affects the active site, still affecting the active site via blockade of access or widening the access, thus resulting in structural change and therefore the activity. Okay, the enzymatic activity lah, catalytic. Okay, so I hope it's rather straightforward. Okay, if you have any question, let me know. And the third type is where you have an uncompetitive inhibition. Okay, so if we looked at one that binds, one that binds to the same active site of the substrate, the other one binds to another site, not the exact same active site, but still changes the active site. And the third one over here is where it binds to the middleman. Okay, in this case, the ES complex. So when you have ES over here, okay, so this is where you have your competitive and non-competitive. It binds to the enzyme directly. Uncompetitive binds to the ES complex. Why can it bind to ES complex? Again, say for example, if you have an enzyme like that, okay, and your uncompetitive inhibitor is something like that. So that's the structure. So if you think about it originally, if you're just looking at these two structures, you can say, well, it can bind, perhaps, okay, but perhaps it binds in a weakly manner. But if you have your substrate over there, going into over here, now you have a nice tightly fit inhibitor. Okay, so in principle it works. In principle it can happen, but whether it happens or not is a different story. But it's very, very difficult to actually identify and isolate this non-competitive inhibitor. Okay, so again, an inhibitor inhibits or slows down or stops enzymatic reaction either temporarily or permanently. Okay, and as I mentioned until today, okay, so I've looked through this morning if I can find any more recent publication about non-competitive and uncompetitive inhibitor. Unfortunately, I do find a few, not as much as a competitive inhibitor. So competitive inhibitor is very, very kind of like straightforward, and that's how normally drugs are designed. So especially antibiotics. So antibiotics normally they are designed to be a competitive inhibitor, especially the original ones. Of course, you know, you have a variety of different inhibitors that you can actually design. Okay. So till today, there is still lacking in providing that all non-competitive and uncompetitive inhibitor actually exist. This is because it's very hard to either get it crystallized or to analyze a binding interaction using our current technology. Okay, maybe in the future we will manage to do it, but so far it's still a bit difficult. Okay, does it mean it doesn't exist? Well, no, because we do have proteins in our body. We have multiple proteins in our body that always interact with one another. Okay, so we have thousands or over millions of proteins in each cells, and each of these proteins are actually never working alone. Either, okay, so there must be a control mechanism. Imagine if you have a lot of insulin in your body, okay, and that insulin is always active. So anything, any sugar that you eat, it always converted to glucagon. But you still need to remember that your body needs glucose, which is sugar, right? to actually produce energy. So if insulin in the cell is always active, how can you actually convert glucose to energy? Okay, so there must be something that stops or inhibit insulin from working 24 hours, okay, so that you do have sugar floating around as the energy carrier in your cell. Okay, so there must be a control mechanism, and thus the control mechanism can be either competitive or non-competitive or even uncompetitive. So we do not know, and it is still a field that is being discovered. Okay. All right, so this is one example of uncompetitive inhibition. So this I found this as part of the journal. So what it does is, if you look at the structure on the right, on the left and the right hand side, so this is actually the same enzyme, but over here you have a different inhibitor, so to say. Okay, if you are interested, you can just go and well, you probably have the link already. If you are interested, then you can go and read the paper. Okay, this is just to show you an example. By no means this example, this specific example will come out in the lecture. This is an example. Okay, oh sorry, not lecture, exam. Okay, similarly, this is an example of uncompetitive inhibition. Oh, non-competitive inhibition, sorry. I think, yep, I have yet to find uncompetitive inhibition. So we do have, this one is competitive, very general. So meaning that the inhibitor is fighting for the same active site. Okay, and non-competitive inhibitor is where your it binds to a different site. So if you look at this structure over here, okay, so this is a crystal structure of cyclohexylamine, inhibits alkaline phosphate phosphatase activity in a non-competitive way. So right image. So you can see here is where you have the the active site of the enzyme. Okay, so this is the natural substrate for this particular enzyme. But when you add in cyclohexylamine, you can see that the inhibition is not exactly on the active site. You can have it there, you can have it here as well. Okay, so this is one of the rare example of a non-competitive inhibition and how it works. Okay. All right, I think that is all for today. These are some of the references, protein data bank, the crystal structure, some of the images that I reconstruct from protein data bank, okay, especially for the non-competitive inhibition. These are some other references on where you can find protein information, additional reading materials, creative biostructure. This one is for the affinity chromatography. Okay, so that is all for today. Very brief, I would say. Even inhibition itself can be very detailed, but we did not cover every single thing. Okay, so this is just kind of like a rough idea for you guys on the technology that is available. All right, so next week we will look at in vitro, in vivo technology, okay, on how you can actually, or some examples perhaps, on how you can actually use cells as the factory for a specific reaction. Okay, that's all for today. I will post the next week lecture notes by this Friday. I didn't manage to post this. Yeah, so once it's ready, just have a look at Spectrum and you should be able to find it. That's all from me for today. If you have any questions, just let me know. We have a WhatsApp group. Okay, otherwise that's all. Thank you and Happy New Year.
Thank you, Doctor.
Happy
Thank
New
you,
Year.
Doctor.
Thank you, Doctor.